Hi everyone. In this webinar, we will talk about one of the advanced sequencing technology, single-cell sequencing. We will mostly focus on its workflow and application. Hope you find it useful. In this webinar, we will begin by providing a general background on single-cell sequencing, exploring its significance in human biology. We will then walk you through the workflow of single-cell sequencing, including single-cell isolation, sample prep, sequencing, and data analysis. Next, we will talk about some real-life applications of single-cell sequencing in research and clinical practice. Finally, we will end with the discussion on the future direction of single-cell sequencing. The human body is an intrinsic system of trillions of cells, each with unique characteristics and functions that collectively drive both health and disease. However, even within a single tissue, cells show great diversity, with distinct types and states contributing to its physiology and pathology. Traditional bulk sequencing methods, which pull genetic materials from a large population of cells and provide a blended view of cellular information, masking its variability and diversity. This limitation is particularly obvious in complex conditions like cancer and brain disease. Single-cell sequencing overcomes this challenge by capturing genomics and transcriptomic data at the individual cell level. To illustrate its significance, think of bulk sequencing as analyzing a fruit juice blend. You may detect general properties like sweetness or acidity, but lose the distinct characteristics of each fruit. Single-cell sequencing, on the other hand, allows us to examine each fruit individually, revealing unique flavors, textures, and nutrients. After hearing what single-cell sequencing tells us, let's now think about the question, why do we need single-cell sequencing? As we mentioned earlier, an organ or local tissue typically consists of many different cell types. To make this concept more intuitive, think of a tumor as a basket of fruits, as shown in the left, with each type of fruit representing a different cell type in the tumor. Even though within the same cell type, like they're all strawberries, you will notice from the graph on the right, they can vary in shape and appearance, just like individual cells within our body. To summarize, in complex tissues like tumors or brain, different cells can exhibit diverse functional states, even if they appear similar in bulk sequencing. Single-cell sequencing enables researchers to identify mutations and markers present in rare cell populations, including stem cells, immune cells, cancer subclones, and the heterogeneous gut microbiota. By combining with other omics technologies, single-cell sequencing results can be used to generate an integrated map of all cells across the human body. Let's now move on to single-cell sequencing workflow. There are many different single-cell sequencing protocols developed by different companies and researchers. In general, the workflow includes single-cell isolation, library prep, sequencing, and data analysis. Let's start from single-cell isolation. There are many ways of generating single-cell conditions. The first single-cell sequencing was performed in 2009, and the cells were separated manually. As you could imagine, it was not efficient at all considering how many cells you need to process in order to produce meaningful data. One of the high-throughput methods of separating cells is fluorescence-activated cell sorting (FACS), which uses fluorescent markers to label cells based on specific markers. And then the labeled cells can be quickly separated. Recent advances in droplet microfluidics provides an ultra-high throughput way of generating single cells in individual droplets that can be then sorted and processed. Here is an example of a microfluidics station at the Lab Lab from the Biomedical Engineering Department at the University of Toronto. You will first need syringes to deliver the sample into the microfluidics device, and some syringe pumps to exert force on the syringe and pump the sample in. And finally, the microfluidics device that will separate individual cells and encapsulate them into droplets. Here is how the droplets are formed in real life under a microscope. A major advantage of using microfluidics is that we're able to generate more than hundreds and thousands of drops. And at the same time, cells will be encapsulated into the drops without the need of knowing specific markers on the cell. Compared to other microfluidics-based methods where people do laboratory prep outside the droplets and then encapsulate the prepped material, droplet-targeted amplicon sequencing DoTA-seq is able to perform all the nucleic acid extraction and laboratory prep reactions within the droplets, which simplifies the whole process. Considering its versatile uses in genomic research and simplified workflow, this webinar will focus on DoTA-seq, which can be applied to both DNA and RNA sequencing. It is designed to study highly heterogeneous cell populations, meaning that it allows scientists to analyze many individual cells at once to look for specific genetic differences.
Let's now look at some details in the protocol, starting from a cell population that is highly heterogeneous. A sample containing the cell population will be prepared in hydrogel solution. Running it through a microfluidics device will generate hydrogel droplets containing single cells. This is what it looks like under a microscope. Then the hydrogel droplets will solidify and form a hydrogel matrix once it is mixed with hydrogel precursors. DNA will then be extracted. As shown in this figure, genomic DNA has been extracted within the drop and visualized using cyber-grain staining.
At this stage, we have extracted DNA from individual cells, and the DNA belonging to that specific cell is in the droplet. The key advantage of doing single-cell sequencing is that we're able to learn the genotype for each individual cell. To achieve this goal, we need to attach a unique barcode to the extracted DNA, and each droplet will have a different barcode to help us to identify the cells from one another. To add the unique barcode and sequencing adapters to the DNA fragments within the droplets, we will use the microfluidics again to generate drops that contain the solidified hydrogel from the previous step. If you take a closer look at the droplets, the droplet formed here contains the hydrogel that contains the extracted DNA. Surrounding the hydrogel, it is the PCR mix that contains the unique barcode, design primer, and sequencing adapters.
Then through multiplex PCR, we will be able to build the amplicon, i.e. PCR product, by attaching the primer, sequencing adapters, and barcode to the DNA fragment with a unique identity to that specific single cell. And now we can break the droplet and pull all the amplicons together and send them for sequencing. For sequencing, many platforms are available for single-cell sequencing, including third and second generation sequencing platforms like PacBio, Oxford Nanopore, and Illumina sequencer. DoTA-seq usually produced 100 to 200 base pair long reads, so Illumina platform is usually used. Once we get the raw sequencing reads, we first need to do quality control to check the accuracy, coverage, and potential contamination. Then we will process the reads by trimming off identical adapter sequences, and then we will map the processed reads to the reference genome. There are many alignment programs available for sequencing reads mapping with a reference genome and assembly without a reference genome. In order to do data analysis, we need to better understand how we assign the sequencing reads back to each single cell. Let's take a closer look at the read composition. This is a simple schematic view of the processed reads. In general, it has two parts. The first one is a sequence of interest. The second part is a cell barcode that is unique to each single cell. Each droplet contains a single cell, and the genetic material from that droplet is labeled with a unique barcode. So we can trace the reads back to where it came from based on this unique barcode. Basically, all reads with the same barcode originated from the same cell. After the single cell grouping, finally, you will be able to do all kinds of cool downstream data analysis and use it for different applications.
Let's see if we're now familiar with the single-cell sequencing workflow. What is the role of microfluidics in the sequencing workflow described in this webinar? A. To create a suitable environment for heterogeneous cells to survive. B. For metagenomics sequencing. C. For single-cell encapsulation. D. For raw sequencing reads trimming. The correct answer is C, single-cell encapsulation. Now let's talk about the application of single-cell sequencing. Single-cell sequencing can be used to detect the tumor microenvironment, which is crucial in the development of cancer therapy. We will look at an example of single-cell targeted RNA sequencing in acute myeloid leukemia research. In this research, the single-cell technology is used to study the change of tumor microenvironment between relapse and remission-associated acute myeloid leukemia. As a result, the scientists are able to identify genes that are specifically associated with the acute myeloid leukemia, and these genes can be further investigated and used in the cancer diagnosis and prognosis in the future. In this figure, we see differential distribution of cell types between relapse and remission-associated samples. For example, the relapse samples have more T cells than the remission stage. In the same figure, we can see here that the remission-associated samples have more monocytes and macrophages than the relapse-associated samples. These results show the difference of tumor microenvironment between patients who recovered and patients who suffered from relapse. These data may be used as a sign to help identify when a condition is improving or worsening. Through further downstream bioinformatics analysis, the heat map results shows that relapse samples have more exhausted T cells, while remission samples have more naive T cells. These biomarkers identified from the single-cell RNA sequencing from remission patient samples and relapse patient samples can be further studied to predict cancer outcomes in the future. Single-cell RNA sequencing has uncovered unique patterns that can enhance cancer diagnosis and help predict relapse in this case. By combining this approach to study the tumor microenvironment, researchers can better understand how tumors grow and develop. This knowledge could lead to potential therapies to prevent relapse. The future of single-cell sequencing holds great promise across a wide range of fields, from advancing our understanding of microbial diversity and ecosystems to revolutionizing cancer research and precision medicine. This technology is reshaping the boundaries of scientific discovery. Innovations in studying tumor evolution, immune interactions, and rare cell populations will bring us closer to breakthroughs in treating diseases like cancer. At the same time, its applications in environmental science, biotechnology, and real-time diagnostics continue to grow, offering solutions to some of the world's most pressing challenges. Some of the key areas of development, such as improving sensitivity, integrating multi-omics approaches, and scaling high-throughput methods, will further expand single-cell sequencing capabilities. While technical challenges remain, such as uneven genome amplification during library prep or data interpretation during data analysis,
Ongoing advancements in sequencing technology and computational tools will eventually overcome these hurdles. Single-cell sequencing is not just a tool for understanding life at its most fundamental level, it is a gateway to the future of science, medicine, and innovation. The possibilities are endless, and the journey has only just begun. We hope you enjoyed the webinar and find the information useful. Thank you for watching.